lecture test Monday? Like with
Yes,
the
you could.
That way we can get the grade, and then if we need to take the final
Monday.
on Tuesday.
Let her do it.
If you want to, you can do that. Yeah. Yeah, you could. Yeah, you could if you want to. If you're prepared to come take it Monday if you want to.
Because it's Tuesday anyway, so.
Yeah. And you are done with lab, so you have time. Sure.
So we can do the semi on Wednesday?
Not Wednesday.
We can do it on Wednesday?
Yeah. Yeah. So. Yeah. Huh?
We'll help you grade them.
No, thank you. Yeah. All righty. All right. So done with this diagram or no?
Yeah.
Done? What's going on? Some of you are saying yes, some of you are saying no. We started. Okay. All right.
You're asking us questions.
All righty. I wanted to show you a couple of other things already. All righty. All righty. All right. All right. Some of you were here yesterday and you saw me finish this entire chapter in half an hour.
What?
Yeah.
I missed that. Yesterday you finished.
Yesterday, this entire chapter was finished in half an hour. Yeah. That's okay. Let's not go there. Okay. Yeah. Alrighty. I'm going to show you a couple of other things. Alrighty. La 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 la. Okay.
Paper.
I gave you some data when we first started this chapter. How much antibiotics are used in the United States, right? All right. Anybody remembers those numbers? Exactly. So because we are videotaping this today, so just for the no, you don't have to really. This paper was published this year. This is the latest. So about 30 million pounds, right?
Yeah.
Look at this. Out of this 30 million pounds actually go to animal feed, okay, and only about 8 million, okay, goes to treat human diseases, okay. And I have another paper that tells you the distribution of why these antibiotics are used. Okay. Therapeutic versus subtherapeutic versus therapeutic use of antibiotics. Why do they use antibiotics? Increase the daily body weight of the animal, improve the food to weight ratio, increase the voluntary intake of the food, decrease both illness and morbidity rate of the animal. These are some of the major reasons farmers add. And then many popular fast food chains have issued a statement that they will not purchase meat from suppliers who engage in the subtherapeutic use of antibiotics. Ha ha ha, yeah, right. Hardee's, the salsa, French Mexican, like KFC, the McDonald's and all that. Do they follow their own oath? Yeah, right. Yeah, sure. This is just a piece of paper. They do it anyways. Yeah. So this is all politics. So they do it anyways. So they don't follow their own oath. All right. Just wanted to share that with you. Let's come back to our lecture. Okay. So we were talking about, last time we left, we were talking about the different modes of actions of antibiotics. The first one we already finished. One of the 5 targets that we talked about last time where antibiotics work was cell wall synthesis. Okay. Inhibition of cell wall synthesis, Today we are starting with the inhibition of protein synthesis. Okay. All of these antibiotics, they inhibit protein synthesis, but their targets are different. Their mode of action is different. Generally speaking, eventually they all stop protein synthesis, but their target is different. How different? Let's look at that. Okay. So I asked you last time, or maybe I didn't. Okay. If you look at this diagram and tell me which of these antibiotics interferes with the process of transcription. Did I ask you that? Okay. And the answer was? None. Because this is the process of translation. Translation, right? Chloramphenicol, okay, attaches to the 50th portion of the ribosome, okay, and does what? Prevents the bonding between these amino acids. In chapter 8, when we were talking about protein synthesis, remember when we said that these, when the ribosome moves forward, okay, the old tRNA moves away and a peptide bond is made between the new and the old amino acids, okay, and the naked tRNA is released, right? In the presence of chloramphenicol, there will be no peptide bond. Why? Because the enzyme that glues them together called transpeptidase that comes from 50th portion of the ribosome. So chloramphenicol stops the synthesis of transpeptidase. Okay, that's the function of chloramphenicol. Stops the synthesis of transpeptidase. What does that do? Okay, portion inhibits the formation of peptide bond. How? By eliminating transpeptidase enzyme. No transpeptidase, no peptide bond. Okay? Tetracycline. Tetracycline interferes with the attachment of tRNA to mRNA. Okay? No bonding between the codon, which are located on mRNA, and anticodon that are located on tRNA. tRNA. This is the one that many of you missed on your quiz in the lab, because lab it is not elaborated, it's not explained that well. I'm going to fix that in the new edition. But tRNA, on the tetracycline, the difference between the mode of action of tetracycline and streptomycin, which will come later, is this. Tetracycline stops the bonding between the tRNA that has the anticodon and mRNA that has the codon. Streptomycin is way up over here. Okay, its target is small portion of the ribosome. I'm going to draw my own diagram to explain it to you, because I don't like this diagram here. We'll come to that in a minute. But erythromycin right here. At one time, erythromycin was used very extensively. Nowadays, because many bacteria have become resistant to it, it is not used very commonly, erythromycin. Erythromycin is like, if you go to big cities, Chicago, New York, Boston, or big cities, if you owe city traffic tickets, they will come at the night time or during the quiet period of the time, and they put a padlock on your car. Really? Okay. You cannot move your car. That's exactly what this erythromycin is, a padlock. It jams the ribosome on the mRNA. Ribosome cannot move on mRNA. Okay. Erythromycin binds to the 50th portion and does what? Prevents translocation movement of the ribosome along mRNA. Literally freezes the ribosome on mRNA, like a padlock. Okay? Now the last two antibiotics, gentamicin and streptomycin. Now this handout, you have this in your package that I just gave you. What do these two do? They change the shape of the small portion of the ribosome. These two, they show you there's a little bump right here. Okay? They change the shape of the small portion of the ribosome. By doing that, what happens? They cause misreading of the mRNA. Okay? It's not a very good diagram, but I'm going to draw my own, explain it to you what goes on. Misreading of the mRNA. Let me draw my own diagram and explain it to you. First, the normal RNA or normal ribosome. Normal ribosome. Normal ribosome, small portion, 30S. A, C, G, U. This is your mRNA. Okay. And here's your large portion of the ribosome, which is 50S, normal. Now a person is taking gentamicin or streptomycin. In the presence of streptomycin or gentamicin, the shape of the small portion of the ribosome will be different. It will be abnormal. They are showing a little bump. I'm going the other way. I'm showing it in a little ditch. Instead of going up, little ditch. Okay? Abnormal shape. Now, when the mRNA binds to the small portion, this is what's going to happen. A part of the mRNA will be trapped in this little ditch. Okay? A, G, U. Okay? So the new codon, original codon is AAA, CC, CGGG. So the new codon will be AAA, GUU. Instead of CCC, oh, sorry, I forgot CCC. There will be C, and then G, U. New codon will be CGG, like that. Because part of the DNA, part of the mRNA is trapped in this abnormal shape, so these two C's will be ignored, and the new codon is CGG, like that, instead of CCC, CGG. Okay, that's called misreading of the codon. Make sense? Alrighty. Alrighty. So that is inhibition of protein synthesis. Eventual end result, same thing. All of these, they inhibit protein synthesis. Now the next one is inhibition of plasma membrane. Okay. Inhibition of plasma membrane. Right here. Would you consider an antibiotic that inhibits plasma membrane narrow spectrum or broad spectrum? Narrow? An antibiotic that inhibits or whose target is plasma membrane, narrow spectrum or broad spectrum? Couldn't tell. Narrow, broad. Okay. Okay. Remember, all living cells, in chapter 4, we said all living cells, they have three things in common: cell membrane, plasma membrane, cytoplasm, and DNA. So any antimicrobial agents that target these three things will be considered broad spectrum. Broad spectrum. Okay? So this is broad spectrum. Okay? All right. All right. Absolutely, absolutely, yeah. Injury to the plasma membrane, damage to the plasma membrane. Remember, okay, this is just introduction, and if you remember the function of the cell membrane in prokaryotes or bacteria, you know how important it is, because bacteria do not have mitochondria. They make their ATP, as David said, in cell membrane. So if you interfere with cell membrane, okay, you are messing up the bacterial's ATP system. You cannot make ATP. Your activity will shut down. Okay, so that's what it says. The cytoplasmic membrane is a selectively permeable membrane. It holds the transport of metabolites in and out of the cell. Okay, so any damage to the structure will hampers or destroy the ability to prevent a number of necessary biosynthetic functions. The most important function that takes place at the cell membrane in prokaryotic cell is cellular respiration, ATP synthesis. And if the bacterium is photoautotrophic, then what else is going on there? Photosynthesis. Very good. 3 antimicrobial agents are here. Polymyxin B is antibacterial. Antibacterial, that's important for you to distinguish, please, because it binds to the phospholipids, okay, in the bacterial cell. The other two, nystatin and amphotericin B, amphotericin and nystatin, they will not work against bacteria. If you have bacterial infection and keep on applying nystatin and amphotericin B, why would they not work against bacteria? Because the target is not there. The target, remember the difference between bacteria and fungi? Sterol. The target of amphotericin B and nystatin is sterol, which are found in eukaryotic cell membrane, which is bacteria. That's why they are antifungal, or they work against eukaryotic cell. All right. Now the next two targets, they kind of overlap. They go hand in hand, side by side. And as I mentioned to you last time, especially the last one, okay, inhibition of nucleic acid should be your last choice. You should not take or prescribe or take these antibiotics, antimicrobial agents lightly, because all living cells, they make the same nucleic acid. Okay? They use the same building blocks to make their nucleic acid. Okay? And these are commonly used to treat extremely resistant microorganisms, MRSA, okay? Maybe chlamydia, resistant TB, okay? And mostly viral diseases, okay? By the way, which antibiotic would you use to treat HIV? Exactly, very good, okay? You cannot use an antibiotic to treat virus. They don't work. Why not? Different target, good. Remember, viruses have no metabolism. They don't have ribosomes. They don't have cell membrane. So what do you use? You use antimicrobial agent. You use like AZT is not antibiotic, antimicrobial. Both antibiotics and AZT, they are antimicrobial agents. They work against microorganisms, okay? But antibiotics, they work against bacteria, okay? Antibiotics do not work against viruses because targets are different. Look at these targets right here. They work against living microorganisms, okay? Antimicrobial agents, okay, they can work against viruses too, okay? Let's look at nucleic acid, inhibition of nucleic acid, and the other one, last one, molecular mimicry. Inhibition of nucleic acid synthesis. Any antibiotic that specifically reacts with the DNA to prevent its replication or transcription will inhibit cell growth, not only cell growth, but the growth of the host cell too. Cell growth and the growth of the host cell. That's why the use of this antibiotic is limited, because the lack of selectivity. Not only that it will kill the pathogen, but it will also kill the host cell. Cannot discriminate. And that is still the biggest challenge to the pharma industry, lack of selectivity. They are working on thousands of chemicals, okay? And once in a while they come up with, if they are lucky, come up with one chemical that they call magic bullet. Remember the magic bullet, the term that we learned at the beginning? Magic bullet. Because it has the selectivity. Like Lysol. Lysol kills HIV, right? On table. But unfortunately, you cannot sniff it, Lysol, okay? Lack of selectivity. It will kill the host and HIV, okay? Iodine, rifampicin or rifamycin, what's the other name for, what's the other name for rifampicin? Rifampin or rifamycin, or what's another name for rifamycin? Rifampin. Rifampin? Is it rifampin? Yeah, rifampin. Rifampin, rifamycin, the same thing. There are different names for this. TB, tuberculosis, nilotic acid, trimethoprim. They all stop synthesis of DNA. Enzyme activity, same kind of overlaps with DNA. Eventually they'll all stop DNA synthesis. We did talk about this one in chapter 6 or 5, in 5 actually. Sulfa and PABA, if you remember, right? All bacteria use PABA to make what? Folic acid, which is used to make nucleic acid, DNA. So molecular mimicry, remember? Molecular mimicry. Same principle here. AZT you have heard of, right? AZT. Okay. AZT you can put it right here. Vidarabine is a chemical, okay, which looks like purine. AZT also looks like purine. AZT you can add to this category. Purine analog. AZT looks like purine. I don't know if I gave you this diagram or not, but this is what AZT does right here. Here. Oops, backwards. Here. Okay. This is normal thymine right here. Normal thymine. This is AZT. Okay. Please notice that AZT at carbon position number 2 right here, it has an azido group, a little box right here. Normal AZT does not. Okay. Here's the viral RNA. Okay. A goes with T, G goes with C. That's normal complementary base pairing, right? Now, when this is, as I told you, this is molecular mimicry. You try to trick the virus to pick up look-alike molecule, right? Normal thymine does not have the azido group. If the virus picks up the normal T, replication of the virus will continue. But if by mistake the person who has HIV picks up, is taking the pill, AZT, if the virus mistakenly picks up AZT, boom, no more nucleotides can be attached to this right here. It will block the replication of the virus right here. This is called molecular mimicry right here. It will prevent the addition of any more nucleotides. Incomplete virus will be synthesized. Okay. So because of the azido group right here, no more nucleotides can be attached. All right. So these type of antimicrobial agents are useful in treating viral diseases because viruses multiply much faster than human cells. Okay. I don't believe you have protozoans and helminth in your notes. Nope. No. 7. Drug resistance. Origin? No, before we go to the origin. Where does it come from? Okay, how does it happen? Okay, I think I did ask you this, and I drew my cow for you. I did draw my cow? All right, good, yeah. So, alteration to receptors to which antimicrobial agent bind. We just looked at some of the targets. Let's say this right here. What is the target of chloramphenicol? No, where does chloramphenicol binds? 50S portion right here. Where does streptomycin binds? 30S right here. Spontaneous mutation takes place. No reason. Okay, before we go any further, let me show you this right here, this number. Look at this number right here. Whenever bacteria reproduce, when you put your bacteria in the incubator, whenever they reach this number, 1 per million to 1 per billion, there is no mutagen, there is no X-rays, gamma rays, or asbestos in the incubator. Under normal circumstances, whenever bacteria reach this number, 1 per million to 1 per billion, one bacterium is going to be different from the rest. That's called spontaneous mutation. So let's say when the bacteria reaches that population, okay, the receptor to this chloramphenicol that is located on 50S portion is mutated, is different. It changes, the receptor to chloramphenicol changes, and the shape of that receptor is changed. Spontaneous mutation. There is no mutagen. Bacteria is not exposed to any of the mutations. Would the chloramphenicol work? No. Spontaneous mutation. Okay. So a mutation that leads to a change in the mutation to the receptors to which alteration of receptors to which antimicrobial bind. Of course, it could be induced, but it's just spontaneous, it happens. Or, what else could cause this? Buddy system. 2 bacteria go to the bar. Oh, can I have the plasmid, please? Okay, sure. Okay, sure. They can go really through the plasmid system, conjugation, transformation, and all that. This is one system. Okay, alteration. Alteration could be caused by spontaneous or through getting the plasmid from other bacteria. Okay, mutation that allows the organism to bypass the sensitive step. What does that mean? Okay. Remember, all bacteria, they make their folic acid. True? All bacteria, they make their folic acid for application. Okay. Bacteria use PABA to make folic acid for DNA synthesis, right? PABA, folic acid, DNA growth. That's what bacteria do. Let's say I'm sick, okay, and the doctor gives me sulfur drug or trimethoprim, and I take my first pill. What does bacteria do? I take my first pill, bacteria now sits down on the couch and starts smoking cigar. Imagine that. As soon as I take my medication, bacteria said, aha, fool, smoking cigar. Why? Because the bacteria is not making folic acid anymore. It will shut down this pathway and will start using folic acid from where? From my body. It has evolved through centuries. It will shut down this pathway. I'm not going to get killed. It's just like human beings. We don't make our folic acid. We get our folic acid from our food. Okay? So as soon as I take my medication, metabolic pathway is shut down, the highway is shut down. Okay? It's like those bacteria is standing at the corner of the street. Will work for food, okay? With the sign, okay? So it will start using folic acid from the host. All right? So that's the second way of resistance, a mutation that allows the organism to bypass sensitive step. Third, mutation that changes the nature of protein in the cell membrane. So microbial agent no crosses the membrane, usually acquired through R plasmid. Many of these antibiotics, they need the help of carrier protein in the cell membrane. Cell membranes, they are required for active transport from low concentration to high concentration. So antibiotics are outside in the low concentration. They need to be transported, okay, inside the cell. So they need carrier proteins. Okay. Yesterday I gave an example. Okay. I'm going to repeat the same example. Name a hot model. Who's the hot model nowadays? Someone gave me a name. Who?
Adriana Lima.
Oh, Adriana. Okay, yeah, sorry. Okay. Adriana Lima, whoever she is. Yeah, okay. All right, Justin. Okay, you remember the name. Adriana Lima. All right. So 2 Adriana Limas now. Not 1, but 2. 2 Adriana Limas. Consider them as 2 carrier proteins. Here is a bacteria, and there are 2 Adriana Limas standing in the door, okay, as carrier proteins, okay? And Justin is the antibiotic, okay? Okay? Antibiotic standing outside, okay? Wants to come in, okay? Would he be able to come in if there are 2 Adriana Limas standing? No? I think so. He'll be a happy antibiotic to come in. Okay? I think so. So carrier protein, normal. Okay? Now imagine Adriana Limas turning into sumo wrestlers. Okay, remember, mutation taking place, Adriana Limas turning into sumo wrestlers.
Oh, okay.
Now Justin coming in? Okay. So a mutation that changes the shape of the Adriana Limas. Okay. Okay, so the antimicrobial agents cannot come in. Okay. So shape change is so big, it does not allow the antibiotics to come in. Okay. I did, by the way, someone asked me to pull up the picture of Adriana Lima, so I did pull up the picture. Okay, and we did see for a second, then it just disappeared. Okay. Yeah. All right. Number 4, development of enzyme that destroys the antibiotic. This is one of the most common. All right. You have the picture of this one. Okay. The most common method. This is like a scissor. Bacteria, they keep a scissor in their pocket. Okay. You take the antibiotic, bacteria take out the scissor and chop, chop, chop. They destroy the antibiotic. Very common example. Okay. You take penicillin, bacteria take out the scissor, chop. Okay. Here's penicillin. All penicillins, not only just generic penicillin, the oldest penicillin. Look at these penicillins, the other. Look at these right here. All types of penicillins, they have this ring called beta-lactam ring. Right here, right here, right here. So if bacterium is resistant to one type of penicillin, it is going to be resistant to all different types of penicillins. One right here, this ring. Because one enzyme works against all types of penicillin. Okay. This enzyme is called penicillinase or beta-lactamase. Same thing, beta-lactamase. Okay. Nowadays, what pharmaceutical companies do? If they give you penicillin, they also give you a supplement with it that destroys penicillinase. So bacteria is no more resistant. The name of that supplement doesn't come to my mind. Does anybody remember that? What? Leonilic acid? Leonilic acid. Okay,
That
thanks.
is present in some like
Amoxicillin and all that? Okay, thanks. Yeah. So that's smart. Okay, so we are ahead of bacteria. Okay. All right. So enzyme of this type is beta-lactamase. Okay. Beta-lactamase, penicillinase. Beta-lactamase is a broad umbrella, big umbrella. Penicillinase, a type of beta-lactamase. Okay. Origin, we just talked about that. No reason, spontaneous mutation. Okay. It can be carried from bacteria to bacteria through conjugation, transduction, or transformation through plasmids. Okay. But please remember, antibiotics themselves do not cause drug resistance, right? Now the next terminology that you have right here that I have underlined, it is not under the type origin of drug resistance. It is standalone by itself. Superinfection. What is superinfection? Superinfection?
like if you have one infection and another, like if you have hepatitis B and then you have hepatitis D at the same time, super.
Okay. Very good. If you have one infection, you treat one infection, and on top of that, you get another one. That's good. But how about another one that everybody can understand? Common example. Ladies take antibiotics and they get yeast infection. Is that superinfection? No? Yeah. Ladies take antibiotics and they get yeast infection. What is the number one superinfection in the United States for people that are hospitalized? In elderly patient, let me make it more specific. C. diff. C. difficile, Clostridium difficile. People, elderly patient, when they are hospitalized and they are on antibiotic for a long period of time, we all have C. diff in our intestine, but our normal flora keeps it in check. But if you're on an antibiotic, C. diff grows. If you smell it, you will never forget it. Yeah. C. diff. It causes what? And extreme enterocolitis. And diarrhea, of course, yeah. Enterocolitis. Yeah. And the last overhead of the class. All right. Okay. I don't see any sad faces. Yeah. Yeah. Yeah, sure. Yeah. See, thanks God. Yeah. Okay. All right. So, yeah. This is actually redundant for those of you who are in the lab. We have done this experiment. Sometimes you go to the doctor and the doctor may give you more than one antibiotic. Okay. 3 outcomes. Whenever you combine 2 antimicrobial agents, there are 3 possible outcomes. Okay. The purpose is to enhance the effectiveness of the 2 antimicrobial agents, which is called synergistic effect. Okay. 1 + 1 equals more than 2. Can you name the 2 antibiotics that produce synergistic effect? Very good. trimethoprim and sulfoxazole. Some of you just took the exam. Easiest one, easier. Penicillin, streptomycin. For some of you, it's too late. Sorry. Okay. Okay. See P right here. P, S, PS station. Easy to remember. But it's too late for you, but for the lecture. Yeah. Penicillin, P, and then S, PS station. So you can use this for the exam if you want to. Absolutely. Why not? Absolutely. Sure, you can put PS station. But don't put PS station. I mean penicillin and streptomycin. Yeah. Okay. Antagonistic. Undesirable. Undesirable. This is just a slip of the hand. Physician wanted to write, okay, penicillin and streptomycin, but wrote down what? Penicillin and tetracycline. Okay. All right. Not good. 1 + 1 is less than 2. Okay. And additive, there's really not any benefit for combining these two antibiotics. Okay. 1 + 1 is just 2, simple math. TT, 2 T's equals 2. TT, 2 T's, TT. TT, trimethoprim, tetracycline. Okay. Why? Why do we combine two antibiotics? Minimize the development of resistant strains. Number 2 is the most common reason, accomplish synergistic effect. Number 3, doctors will never tell you, but they don't know, to provide therapy prior to diagnosis, okay, and to reduce side effects. Any questions? That concludes everything.
Charts going to be on this test?
Just the graphs. Primary, secondary, antigen elimination. Okay. Yeah. All right.
You can show us some of the graphs again, Professor.
I have to set up lab final. Sorry. Yeah. Well, how much time I have? Okay, I can do a couple of graphs. Yeah, I can do a couple of graphs, no problem. Sure. Okay. Those of you who want to leave, that's okay. If you want to leave, that's not a problem. Sure. All right. Antigen concentration. Okay. I will ask you actually questions instead of me explaining to you. Okay. So this is antigen, this is time. Okay. Okay, no problem. Okay. All right. So this is actually primary. First time the person comes in contact with the antigen. Okay. This is phagocytosis. Phagocytosis. Okay. This is catabolic phase. Catabolic phase. Processing phase. Okay. And this is release of IgM, primary immune response, right? Okay. In secondary immune response, green line, short catabolic phase, green line is secondary immune response, short catabolic phase, catabolic phase. What type of antibodies are released? IgG. IgG is made right there. Okay, so those are your two antigen elimination curve. Okay. Antigen elimination curve. Then we do antibody titer. And time. Antibody titer. Primary versus secondary. Primary versus secondary. I don't know if it's catching over there or not. And could you check it's catching this part? Okay, all right. All right, thank you. Primary, long processing phase. Okay, letter X represents long processing phase. Let me use a different color for secondary. Maybe black color. Long processing phase. Letter Y represents release of Ig what? M, because that's primary. IgM. Now tell me, why is it going up here and why is it coming down here? Here it's going up and it's down here.
Antibody are increasing over there.
Yeah.
Antigen
Antigen is going down, antibody is going up. Something is being removed from your system and antibody is going up. Bad thing is being removed, good thing is being made. Okay? Yeah. All right. Now tell me, when should I start the secondary immune response to antigen X? Here? Can I start secondary immune response here? Here? Can I start here? Secondary immune response? Can I? Yeah. Okay. Anytime after this line, I can. Okay. This is the anytime you've got this is the time when memory cells and plasma cells are made. Okay. This is the time when antibodies are starting to be made. Right. So this is the time that tells you immune response has started. At the end of processing phase, right here. This is the end of processing. This is when antibodies are released in your system. So this tells you plasma and memory cells made. So after this, you can start your secondary immune response anytime after that. Okay? So we can start right here. Short catabolic phase, high titer, gradual decline. Short phase, short catabolic phase, high titer, gradual decline. Okay? So let's say letter A. Letter A indicates secondary immune response to antigen X. Letter A represents secondary immune response to antigen X. Oh, how about if I make it a little bit simpler and I say small x? How about this? Small x. Is that okay? Or A, doesn't matter really. Okay. Small x represents secondary immune response to letter capital X. And how about small y? Represents synthesis of what? IgG, secondary immune response. IgG. Okay. Okay, let me ask you this. If antigen X was allergen, if antigen X was allergen, what type of antibody was X? Oh, why? One more time, okay. If antigen X was an allergen, what type of antibody is Y? IgE. If it is, my question is, if this is allergen, I'm telling you it is allergen, okay? And what type of antibody is this? And the answer is IgE. Okay? If it is allergen, always induces IgE. What type of antibody is this? If that's allergen, in secondary immune response, what type of antibody would this be? IgE, exactly. IgE. If it is allergen, it will always be IgE. IgE. Always be IgE. If, as long as it is allergen, it will always induce the synthesis of IgE. The end. Okay? All right. Good luck, guys. Very nice ones. Which letter does what? Just like what I asked you right now.
You are not going to combine these two.
Yes, I will. Yeah. Yep. I will. Yeah. Good day.
When is the test? Like, semi-cumulative Tuesday?
You can take it Tuesday or Monday.
And the semi-cumulative on the same